Assalamualaikum. Hi, I'm Ashley Amro. In this video, we're going to talk about the process of genetic engineering. Genetic engineering is the process of manually adding a new DNA to an organism. The goal is to add one or more new traits that are not already found in that organism. In other words, genetic engineering is simply taking DNA from one organism, combining DNA with another organism, and getting a combination about DNA known as recombinant DNA. Example, a gene from human are cut out and transferred to cells of a different organism. DNA. Deoxyribonucleic acid or DNA is a genetic material which is stored in the nucleus. The nucleus is a part of the eukaryotic cell and contains nucleic acids and is responsible in protein production. Small segments of DNA are called genes. Each gene holds the instruction for how to produce a single protein. DNA is the hereditary material in humans and almost all other organisms. It contains instructions necessary for your cells that to produce proteins that affect many different processes and function in your body to maintain an organism. The word nucleus controls and regulates the activities of the cell. Example is the growth and metabolism. And it also carries the genes structures that contain the hereditary information. As you can see in the picture below, DNA is consists of genes. Genes are set of instructions that determine what the organism is like and its appearance. Example is, if both of your parents have green eyes, you might inherit the trait for green eyes from them. And take note, genes are not just found in humans. All animals and plants have genes too. DNA is usually a double helix and has two strands running in opposite directions. In the picture shown on the side, it has a double helix or a double strand running in opposite direction. Double helix means it describes the appearance of double-stranded DNA, which is composed of two linear strands that run opposite to each other. DNA is the recipe for life. It is a molecule found in the nucleus of every cell and is made up of four subunits called bases and represented by the letters A, adenine, T, thymine, G, guanine, and C for cytosine. As we can see on the picture, DNA have four subunits or bases. It is the adenine, thymine, guanine, and the cytosine. The order of these subunits in the DNA strand holds a code of information for the cell. The genetic language uses four letters to spell out the instructions for how to make the proteins an organism will need to grow and live. These subunits or bases hold a code of information for the cell and uses four letters to spell out the instruction for how to make the protein and organism in order to grow. Pairing of subunits. In the double-stranded DNA, the two strands run in opposite directions, and the bases pair up such that A always pairs with T and G always pairs with C. The A-T base pair has two hydrogen bonds, and the G-C base pair has three hydrogen bonds. Hydrogen bond is a chemical bond between the hydrogen atom and an electronegative atom, or it is attraction between a hydrogen atom which is covalently bound to a more electronegative atom or in a group. Exploring a DNA chain. The backbone of DNA is based on a repeated pattern of a sugar group and a phosphate group. The full name of DNA, deoxyribonucleic acid, gives the name of the sugar present, deoxyribose. Deoxyribose is a modified form of another sugar called ribose. Ribose is the sugar in the backbone of RNA ribonucleic acid. acid. As the picture shown, the most obvious difference is that DNA is a double-stranded molecule, while RNA is single-stranded. And also DNA uses deoxyribose, and while the RNA uses ribose. Numbering of carbon atoms in the deoxyribose ring. The carbon atom to the right of the oxygen is numbered 1, and then around clockwise direction to the carbon on the side group is number 5. In the shown picture below, it says that start numbering on the carbon to the right of the oxygen on the ring and work around to the side chain carbon. Attaching a phosphate group. The other repeating part of the DNA backbone is a phosphate group. A phosphate group is attached to the sugar molecule in place of the negative oxygen hydrogen group on the 5 carbon. Phosphate is a functional group or radical comprised of phosphorus attached to 4 oxygen. Attaching a base and making a nucleotide. One of 4 bases, cytosine, thymine, adenine, and guanine, is added to the above structure to form a DNA strand nucleotide. These bases attach in place of the OH group on the 1 carbon atom in the sugar ring. When a sugar molecule attached to a phosphate group and the nitrogen-containing base, it can form a nucleotide. Joining the nucleotides into a DNA strand. A DNA strand is simply a string of nucleotides joined together. The phosphate group on one nucleotide links to the 3-carbon atom on the sugar of another one. Joining the nucleotides into a DNA strand, it means when a string of nucleotides join together, it will become a DNA strand. As you can see on the picture, in every DNA strand consists of nucleotides. And how is genetic engineering done? The process of genetic engineering requires the successful completion of 5 steps. Step 1. DNA extraction. DNA is extracted from the desired organism. A sample of an organism containing the gene of interest is taken through a series of steps to remove the DNA. Step 2. Gene cloning. Scientists are gene cloning to separate the single gene of interest from the rest of the genes extracted and make thousands of copies of it. Step 3. Gene design. Once a gene has been cloned, genetic engineers begin the third step, designing the gene to work once inside a different organism. Step 4. Transformation of gene or gene insertion. The new gene is inserted into some of the cells using various techniques. Some of the more common methods include the gene gun, agrobacterium, microfibers, and electroporation. The main goal of each of these methods is to transport the new genes and deliver them into the nucleus of a cell without killing it. Step 5. Backcross breeding. Transgenic plants are crossed with the elite breeding lines using traditional plant breeding methods to combine the desired traits of elite parents and the transgene into a single line. Genetic engineering compared to traditional breeding. Traditional breeding is effective in improving traits. However, when compared with genetic engineering, it does have disadvantages. Since breeding relies on the ability to make two organisms to move genes, traits improvement is basically limited to those traits that already exist within that species. Genetic engineering, on the other hand, physically removes the genes from one organism and places them into the other. This eliminates the need for mating and allows the movement of genes between organisms of any species. Therefore, the potential traits that can be used are virtually unlimited.